Okay, good morning, guys. Today we are going to start chapter 3, which is on water and its importance in life. And this will lead us into talking about more chemistry. And then our next chapter, chapter 4, will be on carbon and organic molecules. But I wanted to give you a little introduction to water and its importance because we will utilize water throughout our talks this semester. And also you will use water a lot, and your understanding of water will become really important if you ever take like physiology or a cell physiology or biology class. So, all right, so here's a picture of water and oil droplets. And as you can see, oil does not mix with water very well, right? It doesn't dissolve in water, it doesn't disappear when we say something dissolves in water. And so you can see here that oil droplets form these nice circular structures. And this is going to be kind of the basis of what we're going to call cell membranes. Cell membranes kind of do the same thing, but the molecules that make up cell membranes actually have part of the molecule that will dissolve in water and part of the molecule that won't. We're going to kind of talk about hydrophilic and hydrophobic terms you may have heard of before later in lecture. All right, so, big deal. What's water? All right, water is incredibly important for life. Without water, life doesn't exist. And this is why, for example, people who are searching for new life out there, astronomers, astrophysicists who are looking at planets and moons and whatnot and seeing if maybe life exists on those planets, they're looking for water, right? Whether it's frozen or unfrozen, water would be essential for life, at least carbon life, like we are, organic molecules, like we're made up of organic molecules, and those are important to life. And we'll talk about how intimate water is in relationship to life when we talk about kind of chemical reactions and how water participates in those, particularly those that are involved with organic molecules in our bodies. So water is a biological medium in the sense that if you just look at our bodies, you go into our bodies, we're a large percentage of water. We are a vat of water, if you will. And so the blood, the fluids that are surrounding cells, the lymph that's flowing through lymph vessels, these are all large components of water, right? Or water makes up a large component of those solutions. Your cells are bathed in water. So this water provides an environment for homeostasis. We call it interstitial or intercellular fluid, right? It's where all the chemistry happens. Not only that, water actually participates in chemical reactions, as you'll see here when we get through some of the slides. All organisms require water. They have to get a source of water from somewhere. They need that water. You always have to be a certain percentage of water. A lot of people have disorders, or disorders revolve around dehydration, right? So having the right water content to solute concentration becomes very important. Most cells are surrounded by water, as I mentioned, which we call intercellular interstitial fluid. And cells themselves, the cytoplasm, can be anywhere from 70 to 95% water. So the abundance of water is the main source that life is habitable. And we'll talk about some other features of water that allow the earth to be habitable by life. So we got a couple pictures here, water droplets coming up off of the water surface. And notice that there is water that is streams of water that are coming up, holding on to the droplet. And notice this spider is able to balance itself on the surface of the water. And these two phenomena occur because of hydrogen bonding, which we talked about last time. All right, so water's polar, we know that, right? So this is a picture you saw from last lecture. And you know that water is two hydrogens and an oxygen covalently linked together. And this is a polar covalent bond, which means that oxygen's electronegativity, or its pull on the electrons, is much stronger than what you see on the protons. And so the oxygen typically has a partial negative charge, where the hydrogens have a partial positive charge, because electrons spend more time with the oxygen than the protons, right? This gives water all of its properties because it can hydrogen bond with itself. So notice that the hydrogens attract oxygens, and the oxygen attracts hydrogens of other water molecules. And because there are billions of water molecules, or trillions of water molecules, in hundreds of trillions of water molecules, if you will, in a liter of water, then this hydrogen bonding becomes very powerful, because there'll be trillions of these hydrogen bonds. And it's really hard to pull water molecules apart then. Because of that, because of its what's called high dielectric constant and the electronegativity, dielectric constant is just the attraction or hold of electrons that atoms have with very large nuclei with a lot more protons than smaller atoms. It's a great solvent, particularly for polar molecules. It can pull them or gather them or dissolve them into solution. So let's talk about some of the properties. I kind of talked about that it's a good solvent, right, is one of the properties that water has because of this polarity. So strong hydrogen bonding means it's very cohesive and adhesive in its behaviors. All right, so the ability to moderate temperature, and we're going to talk about each of these in the slide. That is, it can absorb heat and also give off heat depending on the environment around it. It expands upon freezing, becomes very ordered and light, and can form ice, which then floats on the surface of water. And it's versatile as what we call the universal solvent. All right, so you can see here we got ice on the top of water source here and a shrimp underneath. Right, the water underneath is liquid, the water on top is ice. And when you slow the water molecules down so they're not moving very fast, the water lines up in a more crystalline expanse. And it also expands because those hydrogen bondings become kind of perfect and stop moving. The water is much lighter. I'm sorry, the ice is much lighter than water, and the ice will sit on the surface. Here's an example of water droplets forming on plant tissue because they're holding on to the cell wall, which is also polar, of the plant. And it gives you these nice water droplets that you see. And so that's an example of what we're going to talk about here next, which is cohesion and adhesion. So collectively, hydrogen bonds hold water molecules together. And this phenomenon is called cohesion. Cohesion helps, particularly the transport of water against gravity in plants. And what I mean by that is if you've ever used a straw on water and you have a wide straw, you can suck water real easily. If you have a very, very thin straw that has a very thin diameter, it's really hard to suck the water through that. And that's because the water's sticking, if you will, to the surface of the straw, all right? And it gets hard to pull it up by suction. But this property, if you put tubes in water, notice that water flows up against gravity, and because it doesn't fall back down because it's sticking to the walls of the tube. So this ability to kind of bond, if you will, hydrogen bond to the walls of tubes allows plants to pull water up very large. You think about a tree, very, very, really high, right, hundreds of feet, against gravity, using cohesion through what you learned probably if you've taken 155 and what we call xylem, right? These xylem tracheids are long straws or tubes within the plant that allow water to be drawn up from the roots. And it's cohesion that allows you to do this. Adhesion is the attraction between different substances. I love this picture here. So you got a shoot of a plant coming up and there was a dew, and notice the water molecules are sticking against gravity. They're not falling right down because they are bonded, hydrogen bonded to the cell walls, which are also polar. All right, so that adhesion is really neat looking. It's a nice little photograph. So again, it's all because of this hydrogen bonding and ability to be attracted, opposite charges, positive and negatives, on the surfaces and within tubes that allow water to move up in plants and to also kind of be held in place against gravity here after a dew. Water also, because of this hydrogen bonding, has very high surface tension, which means that things of certain weight and certain surface area can float on the surface of water, like a paper clip or our water striders that you see on creeks, or even that spider picture I showed you before. Here is a coin resting on the surface of water. And it's because water hydrogen bonds to itself, it doesn't give so readily. And if you can put stuff right on the surface, it has a very high surface tension. This high surface tension also is why it takes a lot to boil water. You got to boil it, or if you drop something in the water droplet and it splashes in the water and the water droplet comes off the surface, there are streams of water that are stuck to it because of the hydrogen bonding. It's really hard to pull these millions and trillions of hydrogen bonds apart. So again, surface area is related to that cohesion, that ability to hydrogen bond. Moderation of temperature. Water has the ability to absorb heat when the air is warmer than the water, and also give off heat when the air is cooler than the water. And it's this ability, again, to suck in a bunch of heat because of the hydrogen bonds, right? The hydrogen bonds kind of allow you, the water molecules start moving around, but it's really hard to break those hydrogen bonds, so you can absorb tons of heat, right? And then as it cools off, the water tends to give that heat off as the water molecules start to slow down. So speeding up and slowing down water molecules that are attached to each other through hydrogen bonding allows water to absorb heat and also give it off depending on the air temperature around it. And that can really moderate the temperatures around large bodies of water. I'm going to show you a picture of that in a minute. So what the definition of kind of what specific heat is, it's the heat energy required to raise the temperature of a unit of mass by a given amount, usually 1 degree, right? And water can absorb tons of heat before the temperature actually raises, because that heat is transferred throughout the water molecules moving around in the hydrogen bonds. And it's all related to that cohesion again. So heat is absorbed when hydrogen bonds break, right? And heat is released when hydrogen bonds reform. So since you have trillions of these hydrogen bonds, and as heat comes in, you break some of the water molecules, hydrogen bonds, and they start moving around faster, which breaks other ones. But as the heat comes in and starts breaking these bonds, it's absorbed, right? And then as the water cools, then you reform the hydrogen bonds, and you have to release that energy as heat. So that's where high specific heat comes from. So high specific heat of water minimizes temperature fluctuations around ponds and lakes, right? And around rivers. And you guys have all seen the steam coming off of large ponds, for example, or large lakes on a cold winter day, particularly if the day before it was really sunny, right? And the water's absorbing all that UV radiation, and it's absorbing all that heat energy. And then when the sun goes down, and in the morning as you drive past it, it's giving off steam, right? Because the water is giving off heat slowly, and the water in the air condenses when it hits that heat. Another way to look at this is to look at temperatures. So if you ever wondered why the temperatures are much lower along the coast of California than the interior, because as the sun beats and hits the desert, the rock, the mountains here of the interior of California, it warms up tremendously, right? It warms up those rocks, warms up the temperature, surface temperature, and the air temperature very, very quickly. Whereas the sun beating down on the Pacific Ocean, that water absorbs that heat and keeps the temperatures low, because most of the heat is being absorbed in the water. And if it cools down at night, the temperature will change dramatically here. It'll go down into the 60s or 70s in some of these desert areas of California, where the temperature will stay relatively close to, for example, 80 degrees or maybe 79 degrees in Santa Ana, because the water then starts giving off the heat that it absorbed from the radiation of the sun. And so temperatures tend to fluctuate less and be slightly cooler along the California coast because of water's ability to absorb heat and give it off. So that has a tremendous environmental impact. Okay, evaporative cooling is something we can also apply to water, which is really important in mammals, right? Oops, sorry. So we sweat, right? Why do we sweat? We sweat so that we can get rid of heat. So when our metabolism increases, say we get on a treadmill and we start running, and our bodies start burning fuel, all right, to maintain exercise, right, through our skeletal muscles, our core starts producing tons of heat. If our body temperature rises too much, that really affects our ability to do the metabolism that muscle tissue and other tissues are doing during exercise. So what do? We start sweating, right? We release water onto the surface. And that heat, we then draw from the core. Our faces get red, our skin gets red, right? Because we're sending warm blood to the surface. And as that warm blood hits the water, the water absorbs the heat, right? And then it evaporates off as those hydrogen bonds are broken. Heat escapes away from our bodies very quickly. And that evaporative cooling, that ability to do that, allows us to release heat. Dogs do the same thing when they pant. You've seen dogs pant really heavily when they're warm. And what they're doing is they're moving that water from their saliva and from their breath out of their bodies as it collects heat from blood vessels that are just under the skin, the inner skin or the epithelium that lines the mouth. And that's called evaporative cooling, and allows us to cool our body temperatures. Okay, we also call water the solvent of life, or the universal solvent, because it can dissolve anything that has charge or that is polar. All right, and so for example, water is really good at dissolving salts, right? And if you remember from chemistry, salts are typically held together by ionic bonds, right? A positive and a negative. The electronegativity is so high on some molecules that it steals electrons or gives up electrons, and one becomes positive and negative, right? And they kind of stick together. And sodium chloride is a great example of this. Now, what water can do is if you dump some salt crystals into water and you watch, those crystals disappear, right? It's like magic, right? They disappear, they are dissolved into solution. And what water does is it comes in and a bunch of hydrogens surround the chloride molecule and pull it into solution. I should say the chloride ion, sorry, not molecule, and pull it into solution, right? They form kind of a hydration shell. The water molecules actually surround the chloride with this hydrogen bonding, and they pull it into solution. Whereas the oxygen surround the sodium ions, the cations, and pull them into solution. And so they get in there and they work and wiggle their way in, and they break apart these ionic connections and then pull these ions into solution, and they disappear. And that's called hydration, or the hydration shell. And so an aqueous solution is one in which water is the solvent. So, our bodies are large inside of our bodies, are a big aqueous solution of fluid, right, that is found in different compartments of the body. So most of our cells are surrounded by interstitial fluid. Blood is a large percentage of water, and the blood cells are bathed in that, right? So there are different compartments within the body that are cells, and a lot of chemistry are happening within the bodies. Water can also dissolve covalent molecules if they're polar, if they have polar regions, if they have charged regions. So another example of this is if you dump glucose into water, it dissolves, doesn't it? And glucose isn't ionic, it's covalently bonded, but you have some hydroxyl groups that are found on sugars, right? So hydroxyl groups, these OH groups will give up their proton to water molecules. They'll be pulled off by the oxygen, and your oxygen ends up becoming negative when you dump it in water, and you get all these negative charges around the carbon skeleton, and that will dissolve it into solution. It'll pull it into solution. Now, also just as important are compounds or molecules that can't dissolve in water, right? And those are called hydrophobic molecules. So hydrophobic substances or molecules that are covalently bonded typically, but the electrons are shared equally, right? So we call them nonpolar. So these are nonpolar molecules or compounds, which means they simply won't dissolve in water. And this is just as important as substances that will dissolve in water. For example, if you look at our skin is made up of, and structures within our body are made up of, hydrophobic substances so that they can form the actual framework or the container, if you will, that holds the water, right? So a lot of our tissues within the body, a lot of our proteins actually are going to precipitate out of water, which is important, so that they can form the architecture or structural components of our bodies. So the water inside of our skin doesn't dissolve out, right? We don't lose water unless we release it as sweat. And also, if you drop water molecule or water onto the surface of our skin, it rolls off, right? And that's because there are oils and other components of the skin that are hydrophobic and form the structural components of our body. They hold our water. If we're the vat of water, the hydrophobic molecules are the walls of our vat, if you will, that hold water into compartments within our body so that biology and physiology can happen in those compartments. Here's an example of oils, right? So you've all put vegetable oils, or if you haven't done this, put vegetable oil or some kind of oil-based a molecule in water, and it forms oil droplets, right? Or you see oil slicks or gasoline on top of the surface of water, right? At a gasoline station, if there's a puddle, or maybe if you've gotten on a boat in a lake where you see these slicks of sheen that are different colors on the surface of the water. That is hydrophobic molecules that are not mixing with the water molecules. So hydrophobicity becomes really important, particularly when we start talking about cell membranes, right, and how they separate the cytoplasm from the interstitial fluid. So a colloid is a stable suspension of fine particles in a gas or a liquid, right? And you find this is a colloid here in black. Also, you might find a bunch of fine particulate matter in fog, for example. It's not just water molecules, it's a bunch of other suspended particles. And colloids are also important in cells. So all a colloid is inside of the cytoplasm of a cell is the accumulation or droplet of something that's found in the cytoplasm that's hydrophobic. All right, to apply hydrophobicity and hydrophilic together, and if we just go back real quick here, sorry. Whoops. So a hydrophobic molecule does not dissolve in water. A hydrophilic molecule does dissolve in water. Hydrophilic means water loving. All right, so hydro obviously means water, right? The philic here means loving, so it loves water. The phobic means hating, is water hating. And so water, a hydrophilic substance will dissolve in water. A hydrophobic substance will not. It's afraid of water, so it runs away from it. All right, let's talk about an amphipathic molecule. So amphi means half or two paths, right? So an amphipathic molecule has polar parts and nonpolar parts, right? And that becomes really important. We call these detergents. So these are also called detergents. All right, so detergents. There's a couple major molecules here that in the body that are really important, that are very important amphipathic molecules. And phospholipids are one of these. So this is a picture here of a phospholipid. Okay, so phospholipids, put it over here. So phospholipids are large molecules that have a polar end. So they have a polar head. You can see up here. So if I use my my laser here, here's the polar end. All right, and this polar end simply means that there are covalently bonded atoms here, or elements that have large electronegativity, so they attract the electrons more often, and they'll have either negative or positive groups on them, partial charges, either that are negative or positive, that can attract water then, right? They can actually attract water. And water can form a hydration shell around the head and pull it into solution. Whereas the tails here of this molecule are hydrophobic and they hate water. And so you can imagine in solution that these guys are going to organize themselves. If you look down here, these soap molecules are these amphipathic molecules. They're going to arrange themselves with their heads out so that they can dissolve in water, and their tails inward because they hate water, and they'll exclude water and just kind of dissolve themselves into almost like an oil droplet on the inside. And if you have a piece of dirt that's hydrophobic, that's sitting on your shirt, you add Tide or a detergent, right? And the detergent attacks the water or the dirt on your shirt by surrounding it with its tails. And then the water molecules will dissolve around the heads and pull the dirt off of the shirt and into solution. And that's how you get these hydrophobic materials off of your clothes. And that's kind of what a detergent does. Now, what these phospholipids will do is they will form a bilayer. And so if you use this picture right here, and they will do this in solution. They will form a bilayer, and I'm just going to draw 3 of them, like this, that will separate this watery environment, we'll call it W1, from this watery environment, which we'll call W2. All right? And so they will form a bilayer with the hydrophilic head sticking out and the hydrophobic tails basically dissolving into one another to form a very thin oil slick. This is the basis of a cell membrane. This basis with this hydrophobic oil slick in the middle, or these tails in the middle, will separate this water compartment 1 from water compartment 2, or what we can call the interstitial fluid, right, from the cytoplasm. And that's what cell membranes do. They separate the interstitial fluid from the cytoplasm completely so they compartmentalize the cytoplasm away from the interstitial fluid. And that's what phospholipids do. They form the essence or barrier that we call the cell membrane around cells. And so amphipathic molecules have both hydrophilic and hydrophobic ends, right? And they can face each other, right? So that the hydrophobic ends touch and the hydrophilic ends stick out. And so you can dissolve part of the molecule in water. And this forms not only the ability to form cell membranes, but it also forms, for example, the ability to carry hydrophobic materials in the blood. Anything that's hydrophobic, there are several hormones that are hydrophobic. For example, steroids. You may have heard of sex steroids, testosterone and estrogen. Those are very hydrophobic molecules, but they travel through your blood supply, right? How do they do that? Like this. They have carrier molecules that are amphipathic. Part of the molecule is charged and part of the molecule is uncharged. And so they surround estrogen, for example, and they carry it. The hydrophobic tails of the estrogen carrier are going to surround the estrogen, and then poking out towards the water are going to be the polar parts of that carrier, and it will dissolve or pull the estrogen in as an oil droplet and carry it through the blood safely, so that it doesn't move around or precipitate out onto the walls of the blood vessel, for example. So we have all these carrier molecules within the body that are amphipathic that can do this. Another example are bile salts. All right, bile salts are used in the digestive tract to emulsify or dissolve fats in our digestive tract. And so these are all examples of these amphipathic molecules. Okay, we've talked about the importance of water in chemistry, right? So water is a tremendously important in the breakdown and building of organic molecules, particularly the polymers we're going to talk about in the next chapter. So when we start building really large carbohydrates and really large fats and large proteins, we're going to use water to build and break the chemical bonds between these, what we're going to call monomers eventually. So water participates in organic chemical reactions. And these are your two examples. They're called dehydration synthesis and hydrolysis. So when you want to add two organic compounds together to make a larger one, what you're going to do is remove a water molecule. You can see you got a proton or hydrogen coming off of this first short polymer, and then you got a unlinked monomer here. All right, and what a polymer or monomer is, polymers are basically really, really, really large macromolecules that are made up of repeating units. The best example are proteins. Proteins are these really long strings of amino acids. I know you've heard of amino acids before. All amino acids kind of have the same backbone, if you will, or the same chemical composition. They differ because they have one group that sticks off of them that's called the R group. So if you've got 26 amino acids, you have 26 R groups, but the rest of the molecule looks exactly the same. And these are called the monomers, the individual amino acids. If these are 3 amino acids here that are bonded together, if I want to add another amino acid here, I simply remove a proton from the short polymer I'm building. Polymer means repeating unit. These are the 3 amino acids. If I want to add another amino acids to make a polymer that's 4, right, that has 4 monomers, then I remove a proton and a hydroxyl group from my unlinked monomer, then I can link their electrons together. And so I attach these, or I share electrons between the 3rd monomer and the 4th, and release a molecule of water. Dehydration synthesis, remove a water molecule. And that's how I form covalent bonds between my really large macromolecules. If I want to remove that amino acid from the same polymer, then I will add the water molecule back, right? So I take a water molecule and stick it in there and break that covalent bond so that the proton's added to 3 and the hydroxyl group's added back to my 4th monomer, and it will become a unlinked monomer. This is called hydrolysis, right? Because I'm using water to break. Hydro means water, lysis means to break. So notice that dehydration synthesis is anabolism, an example of anabolism. Anabolism is to build something bigger or larger. Catabolism is to break something down. That's what hydrolysis is. Every large organic molecule that's built or broken down in our bodies is going to utilize water. You're either going to remove the water to make something bigger or add water to break something down. So water is really, really, really important. It's very, very intimate. It actually participates in chemical reactions. This is why when you eat something solid, you add saliva to it, right? And you add water in the digestive tract and dissolve all of that, because you got to pull all that food stuff into water in order to do the chemistry, in order to break it down, right? Because the enzymes that are working in our bodies, and also those enzymes utilize water to break bonds. We have to dissolve those materials in the water of our digestive tract in order to do hydrolysis, for example. So water participates directly in these chemical reactions through dehydration, synthesis, and hydrolysis. The next thing we want to talk about is ionization of water. That is, water spontaneously breaks down. Now, I will tell you, this is really, really rare. And what I mean by rare, if I look at pure water, only 1 in about 554 million water molecules spontaneously disassociates or breaks down. That's pretty rare, right? That ends up being about 10 to the negative 7th, all right, times, or that happens, 10 to the negative times that one water molecule will break down within a large group of water molecules. But within a liter of solution, this is still common enough that you have about 60 trillion water molecules that have actually disassociated. All right, so you end up producing a pretty good amount of what we call protons and hydroxyl groups. Right, and this is going to form the biological basis of what we call pH. All right, so this is really important. So what do I mean by water spontaneously ionizes? Well, if you got two water molecules next to each other, every once in a while, one of the water molecules will basically draw off. It's hydrogen bonded, but this attraction between the proton and this opposite oxygen is so strong that it actually pulls the proton off and forms what's called a hydronium ion, which is it's got an H3, right? So it's H3O instead of H2O, right? So this hydronium is typically just called a proton or a hydrogen ion, right? Because it's got an extra hydrogen ion, which leaves you with this molecule right here, which is a hydroxyl group. And this one will be negatively charged and this one will be positively charged. Typically, we represent these as OH negative and H plus, right? We don't include the other water molecule. So water spontaneously disassociates into this because of the draw of the proton to the oxygen. Again, this is really hard to do. It's really hard to break that bond, and it only happens, like I said, in one molecule to every 554 million molecules that are flowing around in pure water, right? Which equals out to be about 10 to the negative 7th water molecules in any liter of solution are actually in this state of ionization, of breaking down into a proton and a hydroxyl group. So we got a hydroxide ion, a negative ion, and a hydronium, which is a positive ion. All right, so it's a pretty rare event, but it happens enough in large quantities of water that it becomes really important biologically. So obviously, these become what we call acids and bases. Now, if we're talking about just pure water, the H3, right, or the proton and the hydroxyl groups, they're going to be equal, right? If you're just talking about a pure water solution and this is happening spontaneously, you're going to have an equal amount of protons and an equal amount of hydroxide ions. And so the pH of 10 to the negative 7th of pure water is what we call neutral on the pH scale. And I'm going to show you the pH scale in a minute. But you can add bases and add acids to solution, and you can change the pH scale. And so we're going to talk about that in a water process. So this process obviously is in equilibrium. When we're talking about pure water, it can go back and forth pretty equally. And again, this type of event is pretty rare, but happens enough within pure water that you actually get protons and hydroxide ions. So let's talk about this as far as its implication in biology. So protons and hydroxyl groups become really important in biological situations. Again, we call this association statistically rare, but it happens enough to greatly affect organisms. Changes in concentrations of the proton and the hydroxide ion within cells or within different compartments of the body can dramatically affect what we call pH. Because water does disassociate, we form ions, the body can utilize this and conduct the protons in particular in important metabolic reactions, particularly when we start talking about photosynthesis and cellular respiration. So photosynthesis helps this process by absorbing energy from the sun, which then splits water more readily. It happens a lot faster than just spontaneous disassociation. And what we're going to do is take the protons that we disassociate the water molecules into, and we're going to pass them along proteins, both within the cellular respiration electron transport chain and within proteins that are found within the they're found during the process of photosynthesis to basically make ATP, to make ATP energy. And we'll talk about the importance of that later. What I just want you to realize is that because we can disassociate water, we can take advantage of these ions in metabolism. And we'll learn how when we talk about photosynthesis and cellular respiration. The freeing of protons and hydroxyl groups in pure water are the basis of what we call pH. And so you can, for example, acidify eggs, put them in an acid, right? And you can disassociate the proteins, and then you can cut them and eat them. These are called pickled eggs, right? Some people really love pickled eggs. And so this becomes really important. Proteins tend to change shape if you change the pH around them. And that can be really, really detrimental if you think about the body, right? If I just let pH go wild in our bodies, then the proteins that we have might not necessarily work like they should. But let's talk about pH first so that we understand this, right? So, as I mentioned, about one molecule in every 554 water molecules actually dissociates into a proton and a hydroxide ion. That equals about 10 to the negative 7th molecules, if you will, in pure water that are actually dissociated. And that becomes the basis of pH, all right? So, and you can see here, if we look at the pH scale, you got a pH of 0 to pH of 14, and then in the middle we have 7, right? And if you remember, I just told you 10 to the negative 7th is the number of protons that you have in pure water that have actually disassociated, right? And if the number of protons have disassociated like that, then the number of hydroxide should be the same, right? In pure water, they have to equal each other since you broke a water molecule down. And so you can imagine if I have 10 to the negative 7th protons, then I'm also going to have 10 to the negative 7th hydroxide ions, right? So when these are equal to each other in a solution of water, pure water, then we have a neutral pH of 7, right? Notice if you combine these, 7 plus 7, that gives you the scale of 0 to 14, right? That's where we get the 14 from. We just take the negative logarithm of it, which I'll show you in the next slide. So this is the basis of pH. Now, if I change the number, as you'll see, if I make this 10 to the negative 6, then this is then you're going to have to change the number over here also, right? So let's check this out. Now, if we add acids and bases, what do I mean by that? If I add an acid to a solution or a base to a solution, all acids, as you can see up here, they donate protons. So I'm going to increase the proton number, right? So if I increase the proton number from 8 to 9 to 10, then I have to drop this number, right? In order to maintain the 14, all right? And it's called the law of mass action. All right? If we're going to maintain this number of protons and hydroxide ions, if I add more acid and this number climbs to 8 to 9 to 10, then I'm going to have to drop this down in order to maintain my 14, right? And that becomes important. If I have more protons than hydroxides, then I'm going to have a lot more what we consider an acidic solution. If I have a lot more hydroxides than protons, then I'm going to have a more basic solution. And that's the basis of pH. So what are some acids and bases that we can add to water that will change our concentration of hydroxide and protons? All right, here are some common acids. Hydrochloric acid, very acidic, right? Carbonic acid, this is a strong acid. Hydrochloric acid. Carbonic acid, it's a weak acid. Ammonium, all right, which is NH plus 4, tends to like to give up a proton. And water can give up. We just talked about it. It can break down into hydroxide and protons, right? So acids, they add protons to solution. Bases can either absorb protons or add hydroxide groups, right? So these are common acids. Here's some common bases. Ammonium. Ammonium likes to actually absorb protons. Likes to absorb protons to become ammonium. Sodium hydroxide gives up a hydroxide, which will combine with protons to form water, right? H plus OH equals water. Phosphates can absorb protons, and water can also absorb protons because they can break down into hydrogens and then add, right, that excess proton to the hydroxide groups. So when you typically add an acid and a base together, they form salts, right? So, hydrochloric acid, if you add that to water, is going to form chlorides and protons, and that's why it's an acid, right? A base like sodium hydroxide will dissolve into a sodium cation and a hydroxide anion, and if you add more hydroxides, it will become basic, right? It'll have more hydroxide than protons. If you add a salt, it doesn't change pH at all, right? So if you have a salt that is made up of ions other than hydroxides and protons, then it's a salt. It dissolves in solution and doesn't change pH. It does change the solute concentration, or what we're going to call molarity eventually, or osmolarity, but it doesn't change pH. So only acids and bases change the pH scale by adding either hydroxides or protons. Some molecules can be both an acid and a base, and they're called amphoteric. So amphoteric molecules can absorb both excess protons and excess hydroxides. And so water is an example of this. Carbonic acid is an example of an amphoteric molecule that can actually absorb either excess protons or excess hydroxides. And these amphoteric molecules become very important as buffers, or what we're going to call buffers. So there's a bunch of math that's involved with pH. All right, and I'm not going to go through the math, but there are a couple important principles here. So when you add up in pure water the number of protons that actually disassociates, I already told you it's about 10 to the negative 7th, right? And if one water molecule in every 554 million is breaking down 10 to the negative 7th at any one instance, then there also has to be 10 to the negative 7th hydroxide ions, right? And so those together equal 10 to the negative 14th actually hydroxides and protons that you find at any one time in pure water, right? So you can see negative 14 is the base of our pH scale. And so if I take the negative logarithm of this, I am going to convert this to a whole number, and I also get rid of the negative, right? So if you throw negative, 10 to the negative 7th into this formula here, you got a negative to the negative 7th gives you 7, right? So every time that you add or you change the pH scale from, let's say, 7 to 6 to 5 to 4, or 7 to 8 to 9 to 10, each jump from 7 to 8 or 7 to 6 gives you 10 times more protons or 10 times more hydroxide ions, right? So changing the pH scale by 1 is pretty dramatic, right? It gives you a lot more protons or a lot more hydroxide ions. When you do change 1, if I add more hydroxide to a solution, then I got to drop the number of protons to maintain my 10 to the negative 14th concentration. If I add more protons, then I have to drop the number of hydroxide ions to maintain my 10 to the negative 14, because that is a universal law of mass action. If you increase one, you got to decrease the other, right? And that's the way it works. And that is the basis of pH. If I increase the protons, then I have a lot more protons than hydroxide ions, and my pH is going to be below. It's going to be more acidic. It's going to be below 7. If I add more hydroxide than protons, then my pH is going to go up and become more basic. So 7 is neutral. That's pure water, where you have equal numbers of protons and hydroxide ions. If I increase the number of hydroxides and decrease the number of protons, then my pH goes up to 8, 9, 10, up to 14, 14 being very basic. If I increase the number of protons and decrease the number of hydroxide ions, then I decrease my pH. And so acidic solutions have lower numbers or lower pH than 7. Basic solutions have higher pHs than 7. And it's based on the number of protons and the number of hydroxide ions in comparison to each other. So hopefully that makes sense, right? If it doesn't, please ask. But that's the basis of pH. And as I mentioned, pH is very, very biologically relevant. So here is pH of some fluids. So to give you an idea, typical body fluids are around 6 to 8, right? So they revolve around neutrality, right? Or the neutral mark. Physiologic pH, when you get into a physiology class, most body solutions are this. Most interstitial fluid, for example, in the body, anywhere you go, has a pH just above 7, so it's just slightly basic, about 7.4. And you eat a bunch of stuff that can be very basic or very acidic. So notice milk down here is very basic. Vinegar, wine, Diet Coke is very basic. Lemon juice is very basic, right? There are certain body fluids that are really, really acidic. For example, stomach or gastric juice is really low on the pH scale, which is about 2. Anything that's very acidic or very basic is typically very corrosive or tends to eat or destroy or damage, if you will, tissue, right? Or dissolve other things. Is really good cleaning agent. So we take in or drink a lot of, for example, cola or like Coca-Cola. Some people like Diet Coke or Coke Zero or whatnot. And the pH is really low of that stuff. And if you drink a lot of it, think about that. You're adding a lot of acid to the body. How does your interstitial fluid maintain homeostasis of 7.4, right? Because that's what your body wants to maintain, is this 7.4, because then all of the macromolecules in your body maintain their shape. If you drop down to a physiology of 7 or just under 7 or above 7.8, you can go into a coma, all right? And so you go into a coma and you can die really quickly from acidosis, for example, if it drops below 7. And so your body does everything that it can to maintain that 7.4 pH. And I'll explain why here in a second. So this is a homeostatic condition that's heavily monitored. And you have a bunch of buffers in your body, a bunch of what we call amphoteric molecules that help maintain changes in pH. So your solutions, your interstitial fluids, the blood, the lymph, any fluid in your body has a buffer system. And that buffer system helps prevent changes in pH because it can absorb large amounts of protons and hydroxide ions. And it does this through using amphoteric molecules, particularly carbonic acid, which I'm going to show you in a second. Remember, going up and down the scale by 1 increases or decreases protons by 10 times, right? So for example, just so everybody understands, a pH of 3 versus a pH of 6 doesn't mean that you have 3 times, right, less or more hydroxides, right? It means you have 1,000 times more or less, because it would be 10 times 10 times 10, right? To go from 4, 5 to 6, that's 3 numbers, right? It's not just 3 times, it's 1,000 times. I mean, think about that. That's pretty incredible. So you can have 1,000 times more protons, or you can have 1,000 times less, and that dramatically changes the charge of that solution and also the properties of that solution. When I say charge, that means the number of positives or negatives you're adding or taking away, right? Because protons are positive. Okay, so let's talk about the importance of pH, right? So pH is important because it can change the shape of some of the really large macromolecules of your body. And I'm going to kind of give you an example of how and why this is important. Our body fluids, which obviously have evolved with a physiologic pH, most of them about 7.4, that means proteins and other really large macromolecules are going to form three-dimensional shapes in that solution that are based on charge, positive and negative charge, and hydrophobic interactions. All right, so let's talk about that. So if we look at an amino acid, we'll use proteins as our example. This is what an amino acid looks like right here. All right, an amino acid has a carbon, an alpha carbon right here in the middle of it as its central kind of atom. And notice, if you remember, carbon has 4 electrons that can form 4 different covalent bonds, right? Now with amino acids, notice you have an NH2 end and then what's called a carboxyl end, all right? A carboxylic acid, all right? So COOH, right? So this is the acidic end and this is the basic end, all right? What do I mean by that? That means if I put this in water, this is what happens. When you put it, if I dissolve this in water, what's going to happen is, remember, water also is always splitting, right, into a proton and a hydroxide, right? Notice what the NH2 does. It absorbs one of those protons and becomes positive. That's the basic end because it absorbs protons, right? Where this gave off a proton, didn't it? It becomes negative. So, you know, a lot of chemists will say that the proton comes off here is added to the amino end. And so you get a positive and negative in solution. This is called a zwitterion, is when you have both a positive and negative group, right, as part of your molecule here, or your compound. And this dissolves in water very readily because it has a negative and positive side, right? Now, R groups, which are here, also have charge, right? They can be charged or uncharged. And if I tell you there are 26 amino acids, I know they're all amino acids because they all have this setup, where they have a basic end, an amino end, a acidic end, or a carboxylic acid end, and then they have a proton, right? The other group is called an R group. So what does that mean? R groups can be different. If I say there are 26 amino acids, there are 26 R groups, right? That give you the 26 different types. Some of these R groups, if I come up here, some of these R groups can be positive, they can be negative, or they can be nonpolar, right? And so what that means is if they're positive or negative, that's going to help dissolve these proteins in solution. Because what happens here is, what I want you to notice is, when you try to put 2 of these amino acids together, here's amino acid 1 up here. Here's amino acid 2, right? When I want to put them together to form a larger protein, I'm putting the monomers together, the amino acids, to form a protein. Notice what happens is it's the amino group of one and the carboxylic acid group of the other that participate in the chemical bond, right? So now I'm removing that charge, that negative charge and that positive charge here, right? And making a covalent bond. And so now notice we're adding the R groups too. If I continue to add this out, I'll have, 10, 20,000, 10,000 R groups, correct? That are sticking up. And so some of these R groups will be negative, some of them will be positive, or some of them will be nonpolar. The nonpolar ones are going to tend to fold the protein up. So you can imagine if I got this long string of amino acids with all these R groups sticking out, let's say there's a bunch of nonpolar ones right in the middle. That protein's going to fold up, right? And so that the nonpolar ends are in the inside of the molecule, and then all of the positive and negatives will be on the outside of the protein. So every amino acid string that you make will fold upon itself to form a three-dimensional shape. And as I've already mentioned, form equals function. The shape of those proteins is really important in their function, correct? So if I change the shape of an enzyme that breaks down something, and I change the shape a little bit, the enzyme no longer works. It doesn't recognize whatever the shape of the compound that it's breaking down, correct? All the proteins in your body have evolved to have a particular shape at 7.4. If I change the pH, you can see from the scale here, from 1 to 11, the number of positive and negative charges changes dramatically. And that's the same on the R groups. So if I have a protein that works in the body, say the small intestine, at pH of 8, works really nicely at pH of 8, it has a certain three-dimensional shape at a pH of 8. If I drop the pH down to 6, the protein is going to change shape or fall apart because I'm going to decrease or increase the number of positive and negative charges based on the number of hydroxides or protons that are found in solution. And if the protein falls apart or breaks down in its three-dimensional shape, it doesn't work anymore. So if I have an overall body pH drop from 7.4 to 7, there are going to be a bunch of enzymes and proteins in my body that are going to change shape and stop working as effectively. And if they stop working as effectively, say either in the brain or the intestines or in any of the organs stop working correctly, and makes you very sick. You can drop into a coma, for example, or even die from that. So acidosis is really important. Maintaining our pH is really important so that the number of Zwitter ions and the number of positive and negative charges on those Zwitter ions, which we call amino acids, are what they're supposed to be so the protein holds its proper three-dimensional shape. Hope that makes some sense. So changing the proteins that are involved with metabolism by changing pH and the number of charged groups can really, really alter our body's ability to survive and do what it's supposed to be doing. Okay, so to prevent pH changes, we have buffers in our body. We have natural buffers. A buffering system is anything that prevents a pH change. And it's going to involve amphoteric molecules, molecules that can absorb both protons and excess hydroxides. Great example. One of the best examples in your body is this molecule right here, which is called carbonic acid. All right, so carbonic acid. This is fantastic. Our body utilizes a waste product called carbon dioxide. All right, so carbon dioxide, CO2. You produce this when you take glucose and break it completely down and basically release all the energy from the glucose molecule and release carbon dioxide. That's what you breathe out, isn't it? Every cell in your body does that. It breaks glucose down into carbon dioxide, right? And water. That carbon dioxide is released. And as it enters the blood, it will basically react with water. All right, it reacts with water to form carbonic acid. All right? So a couple water molecules and carbon dioxide, they will react to form carbonic acid. So your body is using a waste product, carbon dioxide, to produce carbonic acid. Carbonic acid is a weak acid. What that means is it doesn't really change pH that much. It doesn't give up bunches of protons. But it does disassociate, as you can see, into this molecule here and a proton, right? All right, this is called bicarbonate. HCO3 is bicarbonate. It's an anion, right? It's negative. And you got your protons. Now, what's really cool is notice if you get excess, for example, right here, if you get excess protons, right? So you drink a bunch of pop or Coke or you eat something that's really acidic, those excess protons are entering the bloodstream, right, from the digestive tract. The carbonic acid system can do this. It can basically take those. This is what's happening in the body all the time. You have this equilibrium between carbonic acid. New carbon dioxide's always coming in, right? You're making tons of this carbonic acid. Carbonic acid spontaneously disassociates into bicarbonate and protons, right? If you have a bunch of bicarbonate and protons in the body that are equal to each other in the fluid, what can happen is if you bring in an excess group of protons from eating or drinking acids, the bicarbonates will react with that to form new carbonic acid, which doesn't give up its protons so readily, right? It does disassociate, but it's slow. Or if you bring in excess base, you eat something that's really basic, then the excess hydroxides will react with carbonic acid to form water and bicarbonate. So you take one of these and add it to there, right? You get water, and then you get a bicarbonate. So this acid is really awesome. It is a weak acid. It doesn't tend to break down. It does disassociate a little bit to produce two molecules, bicarbonate and a proton, that can actually absorb excess protons and absorb excess hydroxides. And this is one of the most common buffering systems in the body because carbon dioxide is found in all of the solutions. It's found in the cytoplasm, it's found in the blood, it's found in lymph, it's found in interstitial fluid because cells are always producing carbon dioxide. And carbon dioxide is always reacting with water to form carbonic acid. So carbonic acid is kind of found everywhere. It's kind of the way carbon dioxide is carried in the blood anyway, is through bicarbonate and protons. And you can basically absorb excess acids and hydroxides because of bicarbonate and carbonic acid. Some other common buffers, besides bicarbonate, right, are phosphates, amino acids, individual peptides, which are basically proteins. And so proteins and peptides are kind of the same thing. And if you don't know the difference between a peptide and a protein, you will. And when we do our organic chemistry chapter, which is next, they all have the ability to be weak acids and bases and function in the ability to absorb excess protons or hydroxides. Most metabolism in your body happens within a very narrow pH range, and it's the buffers that hold it there. And they're really, really good at this, at holding the pH range between 6 and 8 is really where we need to keep it, depending on what body cavity we're in. And that's what our buffer range is in living organisms, and that's what our last slide shows. Here is this buffering range, if you will. This shows you a pH range, right? And notice this is the buffering range, and notice the pH only really dramatically changes if you're really low or really, really high. So adding a bunch of acid or base really keeps our pH range within this 6 to 4 range in this particular instance, because the buffer is absorbing excess protons and excess hydroxide ions. If you add too much hydroxide ions, obviously you overrun the ability of the buffer to absorb protons. If you add way too many hydroxides, you overrun the ability of the buffer to absorb those. But within the middle range here, which is a really large range, right, compared to your two extremes, your buffer's really good at maintaining its pH. That's how buffers work. They're fantastic. All right, so that ends lecture 2. That ends lecture 3 on chapter 3. We will do chapter 4 next, and I hope to get that up on Wednesday. So have a great day.